Today I have the great pleasure of speaking with Denis Shields, and Denis is full professor of clinical bioinformatics at UCD. And we will chat about in between small drugs and protein therapeutics. What can peptides offer? Hi, Denis. How are you doing today?
Hi, Trish. How are you doing?
Great, thanks. It's lovely to have you here. And first question for you today, Denis, is where are you located and what's been keeping you going over the past few months?
Yeah, I'm here in the Conway Institute. So just outside my door there, it just opens out onto the foyer. So it's nice to hear the sound of human voices out there again as the building starts to come back to life. And I think what's been keeping me going for the last few months probably is just back to human contact. I kind of missed that a lot. I realized teaching is fine. And I didn't realize how much energy you get from students and from researchers when you're engaging with them personally, rather than on one of these little zoomy rectangle things, which is not quite the same thing. And I sing in a choir as well. So it's nice we've finally been allowed to get back. And so that's been good as well, to be in person doing stuff with people. So that's great.
That's wonderful. Denis, you are full professor of clinical bioinformatics. Can you please tell our audience about this role and also what makes you quite passionate about what you do?
Yeah, I think it's all about curiosity, really. I think it's just anything I'm curious about, I get sort of passionate about, because it's just like when I was back in school, I remember learning that life is it's not really, you look at living things, and they look very organic and they grow and they expand and they contract. It looks very analog. But what Sir Gregor Mendel, the monk from the middle of Europe, discovered was that in fact the inheritance of traits is actually digital. It's as units, and he made this startling discovery. And I remember at school being so completely amazed and intrigued by that. And then they went on to tell us about this thing called transcription and translation, which are processes whereby the digital code that's in DNA gets converted into a completely different code that's protein. So there are 4 letters in the DNA code and 20 in the protein code. And these things called polymerases that come in and make the DNA to another version of DNA. And then there's things called ribosomes, make the proteins out of that. And the whole thing I just thought was so bizarre and weird and unbelievable that had to be wrong. So I went to college to discover whether it was wrong. And then now I realized I'm sucked into it. I believe it all too. So it all turns out to be true. So.
Yeah, and so beautifully elegant, I think. So moving on to your title, Denis, in between small drugs and protein therapeutics, what can peptides offer? Maybe you could tell our audience more about this.
Yeah, so peptides to me are intriguing because when I mentioned about the genetic code making proteins are generally quite large in the sense they're tiny to our view, but from a molecular point of view, they're relatively long. They might have lots of subunits in a row. So they could be like hundreds of the subunit elements of the amino acids that are linked together. But peptides are much shorter. They might only be, say, for example, 10 amino acids in a row, 10 of these units that are strung together in a little string. And they're an intriguing space to me, and I'm sort of attracted to them because I think there's opportunities for people who are interested in computational biology to understand more about them. I think it's best to think about this through an example of a project that we're working on at the moment. When the COVID thing came out, people realized quickly enough it was caused by a coronavirus. And so we're trying to do some research now on, can peptides target coronavirus? And one reason we think it's possible is that in the case of HIV, a very successful peptide drug came out called Fuseon. And this drug is based on a peptide, and it was a very important tool in helping keep HIV patients healthy. It's not so popular now because it's an inject-only drug and there are better sort of oral available drugs. But it just demonstrates that when you've got a dangerous virus, peptides might be the answer. And so people are trying to modify the Fuseon type approach. And that's interesting because to the coronavirus, because coronaviruses have a broadly similar mechanism of entry into our cells as the HIV virus uses. And so people have modeled a similar kind of peptide that they've shown some effects in animal models. But our research is really trying to focus on smaller peptides on the basis that they might be a little bit more like drugs, a bit more easy to handle. And so we're just really exploring that space and experimenting with the design of peptides that we make and then get them synthesized by our collaborators in chemistry and then tested in the lab.
That's so interesting, Denis, and we've already questions coming in around that I'm really looking forward to asking you. But our last question then for me is, in UCD Discovery, we are of course very passionate about interdisciplinarity, and I'm wondering, how important is that to your work?
Yeah, it's kind of essential because I'm sort of intrigued by things and I'm curious by things. So I keep on changing my mind about what I'm researching. And the only way you can really survive in research that way is if you sort of lean on the expertise of other people. So, for example, the example I gave of the COVID research, we've got really excellent peptide chemist. I think you probably know Mark Devasel in the College of Surgeons, and so he's been really integral and to that. And we also have a really good virologist, Virginie Gauthier, who's at the testing end of the peptides. So, and then we've got great postdocs who come in, brought different skills into the project, and working then with Dave O'Connell as well here in the Conway, who's a protein chemistry, biochemistry expert. So all together, we're coming together to try and do things. And if I was on my own, you can have an idea on the back of an envelope, but you really need expertise. Otherwise, you make all sorts of foolish mistakes. So it's kind of really essential. And one of the reasons that I think it's really important, and one of the things I've tried to do in UCD is, and in Ireland in general, is encourage training programs for researchers, PhD programs that bring together experimental and computational skills in the same program, so that people have a kind of a blend of sort of experience, which is not always easy because it's hard to learn everything in a PhD, but I'm interested in that idea.
And actually that's something that you've been spearheading, in fact, actually. So you have that's your like a particular PhD network in genomics and bioinformatics, don't you? So
yeah, so there've been various iterations of this. There's one which we started back in 2007, and then Geraldine Butler kicked off one that I was helping her direct, and much more directed at infectious disease, computational biology. And now there's an Ireland-wide one that's kind of spearheaded from Galway, and we've got a lot of UCD investigators who are involved, and we have PhD students from that program based here now in UCD, as well as going all over Ireland as well. So yeah, so it's nice to see the thing kind of growing and expanding out over time.
And connecting people as well is so important. Denis, there's loads of questions. How are peptides made? Do you grow them in a test tube? Can you extract them from nature?
Yes. So there's a brilliant question, because the answer is yes to all of those. You can extract them from nature. In fact, one of the researchers who used to be in my research group has set up a company in Dublin called Neuritas, and they're particularly interested in natural origin peptides from foods, and then using those, because they're so much more like a food process. People will be much safer taking them than they would be taking maybe a chemically synthesized peptide. But you can also synthesize them on a machine. So Mark Devasel in Surgeons, I was mentioning, he has a machine there where he can actually make the peptides chemically. So there's a synthesis that starts on a kind of solid base, and then it adds the peptides one at a time, simply by adding a new tube into the system that then decides which is the next element to get added onto the chain. And then you can also make them biologically by it's essentially genetic engineering. We use this a lot in our foods. I think things like so there are some genetically engineered things in some foods which are not like plants, but in bacteria that produce particular proteins. But you can also get bacteria to make small peptides. It's possible. So there's lots of approaches that can be used.
Okay. And so in our bodies, we take in proteins. And these are made into peptides. Are they the same thing as what you're working?
Yes. So some of the peptides we're really interested in are peptides from the human body. And so we've a collaboration now with Glenn Doherty in Vincent's Hospital and UCD. And we're particularly interested in the whole, how proteins get broken down in the gut, and how the peptides released could have bioactivities, but also they could be an indicator of the process of protein breakdown. And is that going wrong? Is it being dysregulated? Is it going wrong some way that could be affecting disease? So we're particularly interested in studying that. So interested in studying proteins in humans, but also interested in peptides that come from viruses that might be used against viruses. Like, for example, that HIV example I gave is a peptide from a virus that then messes up a virus, if you know what I mean. And then there's peptides which some bacteria synthesize and they secrete, which are antimicrobial against other bacteria. And so we're kind of intrigued at the possibility of using peptides like that, or toxins from frogs or other organisms. These are all possibilities that have come out in evolution. And so when you've got a particular problem, like how do you prevent something like a virus, you might think, well, I might try peptides from all of those sources. And in fact, we are investigating peptides of human source and of viral source and of some frogs and things in our research.
Okay, and so actually there's another question around where do they all fit in to the area of kind of precision or personal medicine, but you've touched on that with gut, but maybe so this is this an idea of kind of measuring those peptides around precision medicine, and everyone's might look a bit different? Is
I'd say it's early days yet. I mean, I think precision medicine is really interesting. We measure things like the genome of a person, and we try to make decisions on the basis of that. That's all the genetic variation that makes us a little bit different from other people in terms of what we've inherited. We can also measure the microbiome, which is just a parts list of all the bacteria that are living in your gut and how many there are of each of those. Peptides, it's really new, to be honest. We're really just starting this work and we don't know where it's going to go. So it's really curiosity. I think it's first of all driven by curiosity, and then there could be applications, because when you start to study things in our body, we don't know what the consequences might be. But at the moment, we don't know if there are any kind of applications like the questioner might be interested in.
Okay, and so here's one. Are peptide medications normally injected, or can they be designed to be easily administered as a liquid or a tablet?
Yeah, they're very rubbish as tablets. And the reason is that our gut is designed as a large machine for digesting various things like proteins, which are peptides, are like parts of proteins. So proteins and peptides, as well as fats and carbohydrates. That's what our gut is fantastic at. So the minute you swallow one of these things, there's loads of enzymes in our gut whose job is get rid of them. There are tricks around that. You can do a chemical trick where you make them sort of reversed of there's things called L and D amino acids. Some of them are, but the D amino acids are non-natural, and they have a different chirality, they have different handedness. And this is really hard to explain because I don't really fully understand it. I never quite get my head around it, but it's an essential feature of chemistry that molecules can be either left-handed or right-handed when they've got enough bonds coming off a particular atom. And so you can make them that way that'll reduce the digestion. But in general, there is an issue of stability of peptides. That is an issue. And we're also interested in things like inhalation or intranasal sprays or dermal patches might be possibilities that might avoid the gut.
Because actually, so for respiratory diseases, for example, an inhalation of a peptide might actually work really well.
Yes, it could potentially be an interesting solution. And there are lots of proteases in the lungs, so you need to take that into account, and patient variability in their protein expression in the lungs. But certainly, it's definitely worth trying.
So here's a very interesting general one. Can peptides really reverse the signs of aging, as so many moisturizers and serums claim?
Well, my assumption is that that's a load of rubbish, because it's in an advertisement. Anything that's in an advertisement is usually trying to make you do something that you don't need to do, that the company wants to do to make money. So I don't really know much more about it than that. I've seen it in an advertisement, so I presume it's not true. I don't know of any scientific evidence that peptides can reverse aging. But
okay. And then I suppose with those advertisements, you always see patent pending. Can actually a natural or any peptide be patented? That's another question that's come in.
That's a good question, and I don't think I can answer that fully. But I think it's potentially every peptide could be patented, so long as you're proving some element of discovery, because I think that's how patent law works. So it's not so much the sequence of the peptide alone would be patentable, but the application that you associate with that peptide would definitely be patentable. That's how my thinking of how to make a patent that's actually got any value would be, that you'd say, okay, I've got this peptide here, I know how to make it, and yeah, that's part of the patent. But the really important thing is I know what to do with it. In other words, I'm going to use it for this or use it for that. We discovered it was for that.
Okay, we're running out of time, so one really quick question, which is probably unfair because it's huge, but I suppose in kind of 15 seconds, untapped potential of peptides? I mean, because they sound really amazing. So, you know, where does the future, where in your eyes? Sorry, only 15 seconds. I'm sorry.
Yeah, the answer is I don't know. I'm driven by curiosity. I think they're fascinating. If you ask me what I'm really interested in studying the distribution of peptides in where proteins are being broken down. But that's just really curiosity about how biology works for me. Where the applications are, I think it's really hard to predict from science. From science, I think it's best done as curiosity driven. Try to understand the world as best we can. And then suddenly solutions just jump out at you once you've got understanding. It's very hard, I think, to go after thinking this is going to be the thing that's going to be great for making a new kind of sliced bread where the slices don't stick together in the freezer. You know, but somebody will come up with that solution with a peptide. And like, why not? And the other thing I'm really interested in, I'm doing collaboration with a company, the Happy Pear at the moment, where we're looking at the distribution of peptides during the breakdown when seeds are germinating, because I think that's a really interesting area, because that's a programmed thing that's deliberately be designed to be broken down into peptides and amino acids by the plant as it germinates. What effects are they going to have on our health? I think all that's really interesting. So I think there are loads of interesting questions, but which will be useful, I honestly don't know. And I'll be fascinated to see that myself.
Amazing basic research. Well done, Denis. And we are completely out of time. So sincere apologies to all of you whose questions I didn't get to, but I just want to take this opportunity before we leave to say a huge thank you to Denis for the most fascinating discussions today. Thank you so much, Denis.
Thanks, Mil, Trish. Bye-bye, everyone.
Thanks so much. And thanks to everyone for joining in. And we'll see you all very soon on Zoom for Thought. Thanks a million. Bye.